How you guys doing? Welcome again to another DNA video. Today we have with us Shayne Morris and Derrick again, the geneticist. Today we're going to do one of the explanations on the actual DNA test itself. So it is a panel that has a bunch of health categories, and they're going to start going through some of the panels to explain to you guys how these panels work, how you can read them. This is part 1 of 6, and we're going to be doing one of these every other Thursday. Today, we're going to be focusing on the cardiometabolic portion and the cellular portion of the readout. Any questions that you guys have, we're going to have the chat up there you can ask questions in. I prefer to put it in the actual Q&A box there, the Q&A box. You can put the questions in there. And then just like last time, if we don't get to it, we will send out an email to you. We'll reach out to you personally with a rep, but we'll get those questions all answered for you. Okay, guys? So I'm going to send it over to the crew and we're going to get started here, okay?
Welcome. Hopefully you guys can hear me. Sorry, I was doing some last minute things, and I got Derrick next to me. He's just off camera. He'll be here in a second. So one of the things we want to do is to help, I guess, facilitate the ease of use of the panel. We realize, there's a lot of data, there's a lot of understanding. Much of that will come, like anything else, when you use a DNA kit and you give it to a patient, and then you start to review the data with the patient. The first couple of times it'll seem maybe a bit overwhelming, but over time, as you do it over and over again, like any other task, as you do it, you learn really quickly, and you get a lot more memory reflex that says, oh yeah, I remember this. And because what you'll find is there's a number of, with the 12 panels we have, and then the sub panels, the traits, that there's overlaps. And when it comes to the approach or the protocol that we need to recover these kinds of genetic predispositions, these variants, really, SNPs, in other words. So it's kind of fun because, for example, in the two we're going to go over today, one is the cardiometabolic panel, and the other is the cellular health. And I'm going to share those with you guys right now so that we can see what that looks like. Hopefully you guys can all see this now. And we're going to review these first 2 panels. And as Matt said, we've got a 6-week project here. So 6 different webinars where we're going to go through 2 panels at a time. And with 12 panels, that's perfect because we will make it through, depending on your practice. So one of the things that I want to emphasize is, as Derrick will delve into some of the more data-driven pieces, I'm going to get him to sneak in here with me. He'll get into the data, best interpretation, and then of course we have the recommender, which we will gradually update and get and bring to your ability. But right now we have it for you when you check in with us based on the data. But one of the fun things about going through this is we have, under each panel, we can go through and not only read the panel, but you can pick out based on the niche you may have. So some of you may have just a global approach to wellness, and your patients come in from anything from, let's say, prenatal care to cardiovascular or pathogens, that are affecting your patients. I mean, depending on where your niche is, it could be detox, it could be performance, and we've tried to cover all those in these panels. And although some of you may not have a ton of clients in cardiometabolic or even cellular health, the data here is still very critical and applicable to patients in the detox realm or the performance realm or the hormone management realm. There is a lot of crossover in the knowledge. We've broken it down for simplicity, but in the end, all of these categories are used to the benefit of every person that takes the test. So to start that off, we've logged in to nbresults.com as a reminder. And once we log in, it'll allow, on the screen there, it says, go to your reports. We went to a report. This is a demo report, obviously. And then once we're in the report, you're introduced to a couple of paragraphs regarding, what your DNA blueprint is about, traits, variants, and SNPs, just a little description. And at any time, if you want to look at the PubMed. So when we get into the report, you'll see first, if we click down on this cardiometabolic panel, gives a brief description of what is included in this cardiometabolic panel. And then, of course, these traits that you see, we have blood clotting, coronary artery, diabetes, heart risks, cholesterol risks, and stroke, and then maybe some sensitivities to compounds. In this case, warfarin sensitivity, which, of course, may also apply to other compound sensitivities. If you want to look even further, if you're one of those people that really wants to see the research behind any one of these traits and or the sub gene names, right? The RSID is here. And then if you go to the DNA, what's it called, Derrick?
At
the very top, it's called My DNA Definitions, which you can see here. This is another report. It looks a lot like your actual DNA report. It does not have the scoring, but it does have the references to the studies that were used to create these actionable genes. And for any of those that you want to deep dive into the research and the references. So that being said, I'm going to close this up first and say, okay, we have a number of genes in here. We don't have time to go through every single trait and cover every panel. And I guess first and foremost, when you open it up, there might be things in here you just don't recognize. So under this first blood clotting genetic profile, we've got, what is that? F11, F5, CETP, KNG1. These are all what we call a gene name. And then the RSID is where we locate the actual single nucleotide polymorphism, or SNP. And then we have a reference, variant, and then what the actual outcome of the person that took the test got. For the most part, I'm going to let Derrick touch on this briefly, because maybe in our future versions of this, we'll rename some of these based on your guys' feedback. But right now, when you have the reference allele and the variant allele, more often than not, the variant allele contributes to the issue.
Yeah, in general, the variant is probably the less desirable, but not always.
Right.
We're discussing ways to make that more apparent.
Yeah, and so when you're reading this, and if you're trying to understand in detail, that the variant is more often the problem variant, but every once in a while there is a gene that the variant is the beneficial one and the reference is actually the undesirable version. If any of that comes up, we're certainly here to help you. But I just wanted to point that out because that question could arise for someone that's really deep diving into their genetic profile. Beyond that, without the deep diving, this is set up to deliver the information where you're simply focusing on the high risk profiles, followed by the medium, and then the low or the normals tend to not need much attention at all. We haven't come across a case where people are using that. It could come up. It doesn't mean it won't arise. I can't say it won't. But for the most part, we focus on the high risk or the high, what's another term?
Priority.
High priority. Thank you. We focus on those. So jumping into this, I'm going to let Derrick take over the mouse because I think he has a couple of genes to kind of have fun with.
All right. So the other thing to say is, even if somebody is medium, but they are presenting with some of these conditions.
Good point.
Like,
yes.
Obviously don't ignore them.
Right.
Just know that they probably still are above average. They're just not super high above average for risk or priority for those conditions.
Actually, I want to thank Derrick for bringing that up because that did come up with the conversations between Derrick and I. This tool used in combination with the clinician's own assessments. So if you have a patient with symptoms and you've assessed certain issues that would genetically fall into the medium, it's not to be ignored, right? The actual interaction with the client or the patient is also as important as the DNA. In fact, it can help guide you in the DNA. For the most part, we see a lot of correlation between the high and also what's presenting.
Mm-hmm.
But when you do have a medium, but the person is presenting with these issues, we can get into some of that later, but that's clearly going to aid you in following through these DNA panels when you are driven a lot by what the patient is presenting. So that's a really good point.
Yeah. And the way that I have kind of been advising right now is that we take your expertise and the information by the DNA and combine the two. Like neither one by themselves is better than them together. So you'll be able to catch things that the DNA is not catching. The DNA may catch things that you can't catch. And so combining those two actions together, you can give the most comprehensive protocol for your client.
Indeed. Yeah, that makes sense.
And we have put a lot of work into the systemic website, the protocol library.
Yes.
So each one of these conditions that are listed on the DNA report now have a list of supplements and directions associated with each one. So if you come across one that you definitely want to focus on, it's now there. And so you can treat that one individually, regardless, or not, that's a bad word, regardless of the what the DNA recommender says.
Yeah. And so that's another resource for you. The first resource is going to be the recommender, which takes the data and categorizes the nutrients and the formulas, right? The nutrients based on the formulas, or formulas based on nutrients, to give you the recommendations based on the DNA. But if you're individually protocoling particular genes, then the protocol library is going to be a good resource as well.
Yeah, exactly. So yeah, today we're talking about cardiometabolic and cellular health. So the scary hard hitters.
Yeah.
Essentially, a lot of these, if a client already has it, they probably already have some sort of pharmaceutical intervention going on sometimes. But there are ways that we can naturally help treat those things. And in many instances, trying to get ahead of it before pharmaceutical intervention is needed is very beneficial.
Yeah.
Like
that's truly the goal.
If you go on a statin, like it's hard to get off of one. But if you can avoid going on one in the first place, you don't have to go through the cascade of side effects that result from going on something like that. So there's a lot here, but if we open them up, let's see. We'll start with some of these heart ones here. This person, you'll notice that they tend to have a lot of the NOS3 gene occurring. This gene is part of the nitric oxide cycle, and it tends to be problematic for people. There's a couple more instances here down here. So don't worry about too much of the specifics on these. We've already done the math for you. So don't feel like you need to understand or deep dive into these things, but know that the NOS3 gene and other nitric oxide genes in this case are they encode the nitric oxide synthase. This is more your
so
knowing how pathways work.
We like this one because nitric oxide synthase is an enzyme that converts arginine into nitric oxide. And nitric oxide, which is different from, by the way, the nitrous oxide that you get if you go to the dentist and you can't handle the pain or you don't want a shot and anesthetic. That's the gas they give people to reduce pain. And that's called nitrous oxide. This is called nitric oxide. And nitric oxide is a hormone. And it's produced from the release of nitric oxide from arginine. So arginine is the substrate. That's the amino acid you want circulating in your bloodstream, so that when this enzyme NOS3 cleaves it into nitric oxide, you get the benefit of creating that hormone. And it's a very short-lived, so it helps regulate vascular dynamics, expansion, contraction, helps regulate many parts of the vascular system throughout the arteries and the veins, but as well as in the heart and in the lungs. It's used in muscle. So oftentimes, if you work in the world of performance and athletes, they use arginine to help stimulate muscle performance. We, of course, work more on the side of the health and wellness approach, where we want to improve heart performance. Now, there's another aspect to nitric oxide that affects men. So nitric oxide it's also the hormone that helps with erectile performance. And so many products out there are designed to support that feature and that function, because it's that vasodilation that allows for male erectile. And if there's a deficiency, oftentimes the arginine can help support that. So these are just a couple of examples of where the enzyme NOS3 performs to keep us healthy. And it's a critical enzyme for all of our cardiovascular system. There's many aspects of it that help us not only with regulating the performance of our cardiovascular, but also preventing the downside of the LDL processes, because LDL is a normal, natural form of cholesterol, but there are instances where that gets oxidized, and NOS plays a role in that as well. So it's an important enzyme, and it's very crucial to our health. In fact, for those of you guys that are here and on this and give us about another month or two, we just finished a research project, an academic one, where we're now submitting the publication on NOS3 with 150 human samples, where we measured the NOS3 SNPs against their role in diabetes, heart disease, cardiovascular heart disease, and Alzheimer's, I believe. And so we're actually publishing on this particular gene. So we have our own real clinical work going on here. So it's not only an important one, but it's important to us because we actually did some research on it that we're pretty excited about, because it was statistically significant, which was fantastic, because this is hard to do in the genetic world, to meet the steep criteria of, we're looking at genes for our nutritional health. How important are they? And in this case, we've discovered that NOS3 is critically important to understand for all of this cardiovascular, and NOS as a hormone.
Yeah, and I kind of chose this gene to focus on because it happens to be in several of these conditions here. It's also kind of neat to see how you can rescue it, like you said, with the L-arginine. Ornithine can also help correct the irregularities in variants for NOS3.
Yeah.
And so we have many of these ingredients in, you said Aventa, and maybe some new ones like Detoxigenic.
Right.
And so it's kind of neat to see how you can go from a problem in your gene, identify that problem, decide which supplement you need to take to correct that problem, and then treat the condition, or support the condition.
Yeah.
So it's kind of a nice little, you can see how the DNA test is beneficial. It helps identify problems that you may be able to see, you may be able to tell this person has high blood pressure, but there are several causes of high blood pressure, or atherosclerosis, or coronary artery disease. So identifying one of the main sources of one of those conditions can help you determine which supplement to use to help correct that pathway.
Yeah.
In a more precise way, I guess I would say, more personalized way.
And that's really, as much as we're going to delve into the nuances of our genetics and these enzymes and other roles that they all play, our end goal is really to deliver you the information, and then provide you with enough solution that you get to make really educated decisions without having to become a geneticist yourself.
Yeah.
So, that's why Aventa was designed, was to tackle the idea that we want to stimulate the NOS3 enzyme, even if it has some genetic variants, that you can still provide enough arginine to boost and stimulate that enzyme for the people that have these variations. So it's really, that is a really good example of many of the things that we're going to go through in all these panels, where nutrients provide the stimulus to overcome these types of DNA variations. And it raises what's maybe functioning at a lower level back to a normal, if not even a higher than normal level. And that's beautiful, because that's how enzymes work. If you provide them with more of their substrate, it makes their job easier to function, even if they're, working at, 60% ability to begin with. We can provide more of that substrate to help them perform better. If not, they're going to stay remaining low performing, and then you're running the risk of, these genetics impacting your health over time.
Yeah. So even if you're not dead, you don't feel great.
Yeah.
Essentially, like you're not running at 100%. You're running at, like I said, 50%, 60%.
Right.
But again, don't worry about having to know how all these genetics work, how the different biological pathways work, and then how they'll interact. We've already done that math for you, essentially. So that's why sometimes some of the results on this report may seem simplified, is because we've already done the hard work for you.
Right.
We've identified which supplements provide the benefits to those pathways and those genes. And we try to list some of them on the bottles. So some of them you'll recognize as treating those genes. But as far as like having to know the nitty gritty, we've kind of taken that part and done it for you.
Indeed. And in the Alimentum products, we have what are called white papers. And all of your sales reps can provide those to you. And those go into a much greater depth on the genetics and why particular formulas and their ingredients support these groups of genes. And a lot of that's written out in the white papers. So even if you're getting ready to do a training in your clinic, or you want to do your own marketing for people to bring in new patients into your clinic, and you really want to engage them in this DNA work, you can use these white papers to your own benefit by discussing potential areas that you can help people improve their health. And that's important for this, for you guys, not only in a marketing sense, but in this validation and ease of communicating this data.
Yeah, and that's kind of the nice thing about these new white papers too, is that they have, I would say probably on average, at least 3, sometimes more references that explain why each ingredient was used. Not just because, well, this other company used it, or that's the way we've always done it, but like, this is why this ingredient's here. What is the clinical dose? We've adjusted a lot of the dosage of some of these ingredients to actually meet the clinical levels. So that's been a high focus for us as well.
Yeah. And I want to take a minute. I know I'm going on a tangent because Derrick just said something brilliant. I'm glad he's here to remind me that oftentimes people are wondering why methylgenic changed subtly from Morse. And I am planning on making videos to go through all these new changes. But for those of you that are here, the concept is when we went through this journey on the DNA, we've increased many ingredients based on the genetic profiles that we were trying to support, like methylation or detoxification. We increased certain ingredients in this new generation of formulas in large part because of a lot of these genetic issues. And that's a great place for me to just take off from that. So back to you.
So the next one to kind of look at, that's people are always kind of asking about, and obviously it's a prevalent thing here in the US and everywhere else as well, is blood sugar, or also sometimes referred to as diabetes. So there are a number of genes that contribute to this. It's very well studied. As you can see, I just keep scrolling. I think there's about 60 or so genes that have been implicated in affecting diabetes. Some of the major ones are ADPOQ, FTO, and PPARG. The names of those are peroxisome proliferator activated receptor, and the fat mass and obesity associated gene. So you can see that one F here. FTO is at the top. ADPOQ down here, and then somewhere in here is these PPARG. So those are some of the classical ones, but there's been a lot more discovered since then. But so just to kind of give another quick little, like, how does this work? In glucogenic, we have ingredients that enhance or correct some of those pathways that break in those variants that help with glucose metabolism or glycemic control to support things like that. I think you can speak more to like some of the
ingredients. Yeah. So when you think of, there's been ingredients that have been used medicinally by natural products practitioners for decades. Fenugreek's one of them. Most of us have heard of chromium being used. And another way to think of that is these ingredients impact either the regulatory pathways, so it could be the glucose receptors. They also impact the ability for certain levels of communication between mitochondria and the membrane receptor that's on the cell membrane. Because of that cell main receptor, and there's, GLUC1, GLUC2, and so on, they transport glucose from the blood and into the cytoplasm, which is then ultimately brought into the mitochondria to finish out creating ATP for us. That is the metabolism of glucose. And that can be interfered with at the level of insulin response. So it can be on the receptor side, because if insulin isn't responding properly, then you're not going to uptake that glucose and it just circulates. And that's, that would affect your blood labs. Those of you that run blood labs, you would see A1C go up, because that's free glucose running around and creating issues with a lot of proteins as there's too much glucose. And A1C is HbA1c is one of the measuring ways to do that. But glucogenic has a number of enzyme, what we call enzyme stimulators. There's some phytoconstituents that stimulate like the transport. And then I would always partner this with what we call energenic, because that helps stimulate mitochondrial. That's a mitochondrial support formula. Because if you want to start bringing in more glucose efficiently and converting it in ATP, the mitochondria has to be healthy. So when you think about this, or when you see a report, you'll see that oftentimes glucogenic and energenic should be used simultaneously to drive up this metabolic metabolism of glucose and improve insulin sensitivity in the process. Now, that's the design behind a lot of these enzymes. And so when you see a gene, many of these are really enzymes and or receptors. There aren't many, I would say, nuclear proteins or transcription factors, but a lot of these are enzymes and or receptors. And those are kinds of things that we can certainly improve with natural products. I mean, nature provides a number of tools that improve our enzyme and our receptor functioning. So that's why this is such an exciting system for us, because the tools have been there for a long time. We've been able to take advantage of the tools regarding the formula side. And now we can apply those same tools very specifically to each and every patient based on their genetics. So instead of, approaching a insulin resistant person with 5 different tools because we want to cover all of the different possibilities, if we look at their genetics first, we can then manage or personalize our approach to that insulin resistance via the genetics, as well as their blood levels and their A1C and so on. We can really start to narrow down our approach and focus on their issues, their outcomes, and their success. And it makes it a lot easier for the clinician. And it also gives that clinician the foundation to fall back on. It's like, look at your genetics. We really want to start here because this is going to be the most impactful place genetically, as well as with, the symptoms and what your blood labs are telling us and so on. So it really starts to stitch together a lot of what we know, but now we have the data to help present to the people, simultaneously. I know that here it's been incredibly, as we are developing this over the years and we test people in the staff and in our families, there is an impact. There's a psychological impact to, wow, I knew I was struggling here. I didn't know why. But now that I look at my DNA, I understand more about why I want to do this journey above and beyond it being a good idea. You tell somebody that they need to stop drinking water out of a water bottle, because plastic water bottles, because plastic can be dangerous for them. When you look at their genetics and you see that their detox pathways are riddled with these polymorphisms and they don't detox as well as someone else, then there's a little bit of pressure on them to stop exposing themselves to these things because they are not going to be able to eliminate or detox as well as maybe their other family member that doesn't have these problems. So it just gives them that extra oomph, because I know it's always hard for all of us to keep people on the path, for sure.
Yeah, the report, I know that a few people that, like you said, we've interacted with, it's kind of like a call to action. It's like, oh,
Yeah,
like,
exactly.
it's like, I know I shouldn't drink plastic out of water bottle or water bottles with plastic. But then when they see like, oh, it's really going to be harmful to me.
Yeah.
They internalize it more, and they start to follow through on things that they know they should be doing, but now they, like you said, they feel more motivated to do so.
That's right.
A couple of side notes we were talking about. So a glucogenic is just the improved form of sinulin.
Yep.
If any of you are familiar with that. And the other thing I was going to mention when you're talking about it is when making these white papers, coming from a purely academic standpoint, classical training like I've had, and now seeing, I was totally oblivious to most of these, like you said, ancient medicines and herbs and stuff. And they're just now starting to research all of them. And it's cool to see,
academically.
Like, yeah, it's like, oh, like, they did this thing in India. They didn't know why. They just know that it worked. Right. Now they're showing like why it works and what compounds are present. And it's kind of cool to see the pieces kind of fit together and be like, oh, this compound in this plant helps repair this pathway that has this broken gene.
Exactly. Yeah. We for years, maybe decades, there's been the anecdotal or intuitive use of these plants, but now we have evidence. It's an evidence-based scenario. And and again, that's why some of these formulas have moderately changed. Glucogenic didn't change very much because that one was dialed in. Others changed more based on some of this genetic information as well as clinical outcomes. So we're always pushing the envelope to move in a more effective direction for all of you. That's our goal. But it was nice to see, Derrick's he's no longer new. He's been here a while. He worked through this entire project as well as the microbiome project. And as a researcher, he's been able to dig into some of these things and contribute to improving these formulas, improving the way we understand genetics, the cell, and our microbiome. So it's really been fun to build these projects for you guys.
So there's more coming in
terms of
these conditions,
particularly this condition. Yeah, in terms of metabolism and glucose, we're bringing new things to you soon.
Okay. So the other thing we haven't talked about yet is the DNA damage or cellular health.
Oh, yeah, let's jump on that real quick. Now we need to caveat this one because this one is a way more complex system, and yet it still needs to be addressed for the main reasons, something that Derrick pointed out earlier. Cellular health in a generic way. We've broken out all the various organelles, mitochondria and energy and detoxification and that. Those are in other categories. Cellular health, as you'll see, has a lot of the cancer. So in terms of avoiding cancer talk, because we don't want to violate any of the, regulatory with prevent, treat, so on with cancer. What we do want to provide, though, is the underlying health issues with any form of cancer or other serious things like this are what we call DNA damage or membrane damage, mitochondrial damage. All of these things are what we call, the core or root causes of some of this stuff. So although we've used these terms cancer, because that's how in the literature it's used when it comes to these types of polymorphisms, as nutritional and natural products clinicians, we want to focus on DNA damage, membrane damage, protein damage, and so on, because those things are very much controlled by oxidative stress. We can use antioxidants, nutritive deficiencies, enzymes that don't work well that repair your DNA. If they don't have their nutrients, they won't repair DNA properly. Therefore, we are concerned about some of the things like NOS. That's in here. You can see NOS2. It's another version of our nitric oxide. The glutathione pathway is critical here. Glutathione is one of the most important cellular antioxidants. Methylation pathways, you can see the MTHFR gene is located in some of these panels. Each and every one of these, although it's defined by a scary term, you'll see that a lot of the benefits of what we can do for the prevention of DNA damage, the prevention of cellular damage, lies in tools that we use on a daily basis. We see VDR, that's a vitamin D receptor. So vitamin D plays a role. Again, I'm going to point out that we have superoxide dismutase. That's an enzyme. We use that in our products. There's so many of these things that are used every day as antioxidants, enzyme support. We have the glutathiones or resveratrol, all of the lipid antioxidants, the lutein, zeaxanthin, beta carotene, and over again. If we get down into, let's see if prostate brings us any new, selenium, right? We use selenium, especially in the methylgenic. There was another methyl pathway. And so here's glutathione. GST is another glutathione pathway. SOD, another vitamin D pathway. You'll start to see this as you guys use this tool yourself. You'll start to see these emerging patterns where, although this panel's big, and at first approach, if you were a patient, you'd say, oh my goodness, I'm so worried about the C word. No, we're here to help you support the cell at every level with protecting the enzymes, protecting the DNA, reducing damage. And how do we do that? Well, the tools exist. We have the detoxigenic, we have the methylgenic, we have all these tools, which of course contribute to the stability of these enzymes that contribute to the health of these pathways. So it's really about DNA damage, membrane damage, mitochondrial damage that leads to these conditions on this panel. And what I really wanted to emphasize here, and I'll let Derrick, chime in, but I want to emphasize is don't be intimidated by the fact that these are listed as bladder, brain, breast, cervical cancers. That is how they're recorded in the literature. That's how the clinical world looking at DNA has connected them, correlated as, and so on. We're interested in the genes here because the genes are controlled by things that we can do, right? We can certainly support the genes that are found in these pathways in a very natural and healthy way. And we're not we're not trying to treat the cancer. We're trying to prevent and support these processes built around this. And so we will learn a little bit more about some of these processes because, as we all know, DNA damage, membrane damage, protein damage, mitochondrial damage, these are things that are going to help us well beyond this particular panel. They help us throughout, right? They help us deal with stress, psychological, emotional, physical. They help us deal with repairing organs and so on. So it's a greater picture, but this gives you a lot of the detail to go over when it comes to cellular health. So really the title of this panel is really where you want to focus. It's all about cellular health. There, you can use the traits to help stimulate the conversation, but really what we're about is the cellular health in this panel.
Yeah, and the recommender goes in and it identifies those products that have those nutrients,
those ingredients
that repair or supplement those pathways. So it's not to say that like glucogenic is going to cure some sort of cancer. It's just saying that glucogenic helps with this pathway that, when damaged for long enough, can potentially lead to DNA damage that could lead to cancer. So it's kind of like trying to get out ahead of it before something goes awry. But as far as information that is in here, we have tried to provide, we'll just go maybe with this one here, the lifetime percentage chance. So that one says 1% of people, even less. One that's more common, breast cancer, 1 in 8 lifetime chance for women of breast cancer. So don't be, just as somebody's listed as high risk, it doesn't automatically mean they have it or they're going to get it. Like if you pop up with a high on ovarian cancer, that doesn't mean to schedule a hysterectomy tomorrow. Okay, we're not trying to be sensational or we're
not trying to create fear and panic.
Fear.
Yeah.
What I would say is, if something like that were to come up high, is to not skip your annual exam, and then again maybe talk with us or look at which genes may be damaged in that one and see what can be done from a nutritional standpoint. But Again, it's not fear mongering or trying to like provide a diagnosis. It's just informational.
Yeah. And DNA damage is a big deal. I mean, we're exposed to more and more things that literally damage our DNA. And all of the, right now we have an overwhelming amount of animal study data and more human data coming, monthly regarding the pesticides, PCBs, plasticizers, BPA, phthalates. These all can be involved in DNA damage, and they are ubiquitous. They're everywhere. So we just need to be cognizant, because we can do things to help really support DNA health, cellular health. It's one of our strong points, is really supporting cellular health. And in this panel, that's what we're all about, is the cellular health.
Yeah,
for sure.
And something like the lung or throat cancers. Everyone knows somebody whose grandma smoked their whole life and lived until she was 95, and then somebody else who smoked for 5 years and died of lung cancer at 50.
Right.
And that's kind of why some people don't seem to be as affected by smoking. And not because, like, I wouldn't take up smoking if I showed up as low.
Yeah, we're not advocating that. Definitely don't smoke. It's terrible. Don't vape. Don't do any of those things.
But yeah, it would be a greater call to action if you had a client who was smoking and then they saw that they were at high risk for lung cancer.
Yeah. Or vaping.
Like it would be good for them to know that like you're at a much higher chance of developing this if you continue with this lifestyle.
Yeah, it's a call to action.
It might be that push that they need to.
I mean, I know vaping has become really popular in the younger generations, so I use the genetic test to remind people, hey, I know you're vaping, and you even probably can admit to me it's not good for you. But when we run your genetic panel, your chances of having a problem are higher than normal. So I would always recommend you stop. But let's say you're at a higher risk. Let's stop now, not.
Yeah. And as far as the verbiage goes on this, low technically means average. So you're at average or normal risk. So it's not, like I said, it's not a invitation to take up certain lifestyles that are associated with these cancers, like because like, oh, I'm impervious because I have low risk. It's like, no, you have normal risk.
Right.
And so,
which is still, it still means we have to make good choices. And of course, in our industry and being in the wellness, I would expect that anybody coming to visit you guys, just like if they visit us, they're aware of the idea that we're not going to accept that it's okay to have, plastic in our plastic water, or vaping, smoking, too much alcohol, processed foods, right? There's a baseline of expectation.
Yeah.
And beyond that, the genes really can help us out. If people continue to choose really bad life choices, environmental exposures, etc., remember that genes are only about 40% of the driver behind, our future health or our present health. 60% of it is still based on choice and exposure and environmental factors and so on. So yeah, it's still super important that each and every person gets that coaching from you guys.
Yeah. If you abuse a healthy pathway, you can break it. So.
You can break it. Yeah. That's a good point.
So those are our two panels. I don't know.
Excellent.
Are we taking questions, or is that just?
I think we're gonna. Let's see. I'm gonna go back to. I can unshare the screen. So hopefully you guys got something good out of that. We'll do that. The next 5 videos will have a similar format, and it'll feel a little bit repetitive, but it's my experience using this DNA, being in the DNA world, that repetitiveness is really helpful, because I've heard it 4 times and I still learn as we go. So don't not join us for these thinking that it's going to be repetitive, because it really is. It really won't feel that way, because everything is so intense and there's so much to do and so much to learn. So that's important. I think we had a few questions. So quickly, Aventa is only a pill form from now. Yes, that's correct. We never did get Aventa in a liquid. However, you guys are the first times I've heard it asked, so it's certainly, we could put that on the to-do list. I didn't realize that that was even a desire. So that's great to hear. Those of you that are moving, that have been patient and are moving from the cellular formulas, the Morris, Energy, Epic, and so on, into the Genix, I want you to be very happy that we are creating liquids for many of those, especially those that liquids are actually very valuable. So methylgenic, energenic, detoxgenic, bindgenic, and so on. They're in process now, and we've actually done some real research on that to give you guys even a leg up on those liquids. They're going to be liposomal. So all of the genic formulas will have a liquid equivalent that is liposomal. So I look forward to start bringing those to you one by one as they come out of manufacturing. You guys are the first to hear that. That's kind of a sneak peek.
And just as a small explainer, many people might not know what liposomal means.
Yeah.
So essentially the potent ingredients are in there are encapsulated in small fat cellular constructs, kind of.
Yeah.
And so they're protected from like stomach acid and stuff until once they hit down to where it's time for fat metabolism, which is where a lot of the absorption takes place of these medicines. So you're not just eating it, digesting it, or just destroying it in the stomach.
It's protected and it's absorbed. They're more bioavailable. The liposome kind of bypasses some of the what we call transporters in the intestines, and they merge and they get transported more rapidly. So it's a more bioavailable, and that's kind of the technology we added to the liquid versions of the genetics, or the genic formulas. Sorry.
And most of them taste pretty good. In my experience.
Not bad.
And I know there's a lot of people that hate swallowing pills. So those were all good reasons.
I know we won't get to all these, but we're always available, and we will try to answer these if we don't get to them. Let's see. Screenshots, fantastic. Yes. And we are making the videos and posting them. Yeah. Let me remind you guys, there is information on the Systemic Formulas website for DNA, some of the videos we've placed there, and you must go to nbresults.com because we've also placed information there. So there's two resources right now you can go to. Anything that isn't there, shoot us an email, talk to your sales rep, because we are still building information every day for you guys. This will be a phenomenal outcome. We're, of course, adding every day, every moment we get, we're adding to it. We're making subtle changes, making it easier for you guys. That's our goal. So we're making a list from, changes that you guys may want in the future. Let's see.
Our treatment protocols for DNA.
Oh, this is a great question. So when we're dealing with DNA, I want to keep in mind that this is genetics. There is another whole other science that we call epigenetics, and that is controlled by a lot of these same mechanisms. It's controlled by methylation, acetylation of the histones of the DNA directly, mRNA interference, and so on. There's all this epigenetics. A lot of that actually is controlled by the microbiome and nutrients as well. And that's the dynamic side. But the DNA itself, so this test, so genetics and these variants, these SNPs, these are permanent. So whenever you get this test done on a patient, it is very important to be clear that any of these panels where you show a higher risk or a higher priority, that is a lifelong consideration. It is not, you don't recover from it. If they manage it, you can manage it both with, of course, we're doing this so you help manage it with supplementation, but clearly the other benefits are going to be diet and lifestyle, like exercise. But it is a lifelong journey. So if somebody is deficient in the methylation pathway, they have to be aware that deficiency will remain their rest of their life. So they have to be very conscientious about their choices forever. You don't actually change your DNA sequencing. This is inherited, and you also give a version of this to your children. So it's always good to know what they got from mom and dad, because their life journey is dictated by their DNA as well. And that is a lifetime journey. It's not you can't change your DNA. So it is something to be aware of, that they have to manage it in some way the rest of their lives to optimize their health for sure. Oh, somebody already mentioned that. So someone did that. Great question. The clear one, smaller capsule. So far, based on the dosing, and I'm looking at maybe it's a really potent formula, and I experimented a few years back with some beta testers on a lower dose, and it was really hard. My only option is to make a smaller capsule with more doses per day or more capsules per dose. That's my only option. If I drop the dose size, then I lose efficacy. There is an efficacy line that I want to be maintained. Otherwise, I don't get the results, or the directive of that product doesn't get the results it needs based on dosing. So, I mean, it is hard. You're right, and the Clear 2 is quite potent in terms of its flavor profile, so hasn't been able to be turned into a liquid very easily as well. It's very potent.
If you're super brave, you could try busting one open, but it's pretty strong.
It's really tough. At the moment, I haven't found a good solution because the dosing is very critical for those two products. Yeah. Yeah, and someone else, I think Sherry Humphreys made a point. You can try to hide it in foods. That works great, especially for younger people. Hide it in foods. I've done that. It can be successful. Sometimes they call you out on it. You get caught. But if you can pull it off, it's fantastic. I think that you guys, so I think that's it. We're going to wrap this up, and we are very grateful for you guys joining. And we're, like you said, we'll do this every other week. Once these are all done, we're going to make a series of videos, and we'll even add to those. And we're going to make a DNA class, and we're going to certify it in the future. So as you're along with the journey, we will catalog this, and we'll end up doing a certification, and we'll even do it in person at some point. So look forward to that as well. Thanks, Derrick.
Thanks, guys, for all that information, and keep in touch, everyone. Like he said, in 2 weeks, we're going to have another one. It's going to be on the detox panel and the energy and performance panel. I am pretty privy to the energy and performance panel because I am really into athletics, things like that. And it's more than that, but it does have a lot of useful information. And keep this in mind. Shayne was saying, remember, the DNA, your DNA never changes, right? Even my friend, I got a buddy, his name's Charles. I told him the moment those kids pop out, let's swab their cheeks. And it's already made, especially for his wife, who's, she's kind of a health nut. She's been really obsessed with looking through this panel and being like, oh my gosh, I can do this. Because a lot of cool stuff you can do, even if you're not sick or unhealthy, that knowing your genes is useful for. So if you order 2 of these tests, you're already at a 3.5% discount on these kits. If you order 3 of them, right, so on and so forth. And if you haven't tried them yet, it's really fun to try it with your family first, right? If you're hesitant, and see how you go about it, because I and the other reps can go through it with you. Derrick can go through it with you. The man on camera himself, you'll get to talk to, or, one of the people that he works with that do the genetics there. So keep that in mind when you're doing this, because as you order these things, they don't have a shelf life on them either. The tests themselves do not. Once you swab your cheek, then there's, I guess you could say, a time limit on that. You want to get that out the door into the lab. But I got a lot of practitioners that stock up on these whole 10 at a time. And, we've seen that if somebody gets converted in the office with it and they swab their cheek, there's an astronomical higher chance that they're going to go through your entire program because they've already done the DNA swab right at the beginning. So it's really kind of a good place to start when you're trying to market a program as well. And so I know we have some more questions popping in, but we're going to keep those questions, and we'll send you, we'll have Derrick or Shayne answer those questions, and they'll send those answers out to you by email, or they'll have the rep reach out to you with that answer. But even if you didn't have a question now and you think of one later too, don't hesitate to shoot an email to info@systemicformulas, or if you know your rep, because we'll answer those questions. But thanks for showing up for this one, and tune in for the next one because it's going to be a lot of fun. Okay, guys? Take care, guys.
Thanks.